Howdy, Pima Aztecs. Welcome to microbiology. This is a brief history of microbiology. This is chapter 1, and I think I have 3 videos for chapter 1, so this is just a micro overview. So let's make sure our pen works. So, as you know, we are animals, Homo sapiens, and animals along with plants and ecosystems in general. So just ecological systems depend on microbes. Most microbes in the world are beneficial. A few microbes are harmful, and those are the ones that get all the airtime because we need to study those to try to mitigate their harmfulness. So this diagram is from the Bauman book. It's the golden age of microbiology, and then also the pre-golden age of microbiology. So let's take a look at this. In the left column, it's the biologists, and then in the right column, it's the modern disciplines that the work of those biologists led to. So this guy, Leeuwenhoek, we'll talk about him, but all the work that he did led to all of these disciplines. So bacteriology, that's obviously protozoology, that's studying protozoans. Mycology, studying fungi. Parasitology, that's studying parasitic protozoa and animals. And then phycology is the study of algae. Carolus Linnaeus led to classifying things, or taxonomy. This is a personal hero of mine, Semmelweis. I taught at Northwest Campus a few years ago, and my students in micro found a Semmelweis t-shirt. So when we talk about Semmelweis, I'll try to remember to wear it, but he was a baller. And then this guy, Snow, led to infection control and epidemiology. So we'll talk about those. And then the golden age, we have probably some of these names you've heard of. Pasteur leads to pasteurization of your milk, for example. Buchner, there's a Buchner funnel named after him. Koch's postulates, we'll talk about that. We don't really talk about Ivanowski, which is interesting because he led to virology, and we're all interested in viruses right now, right? With COVID-19. And then these 2 guys, I'm not even going to try to say their names. Well, okay, now I have to try. Bajornik and Winogradsky. Maybe I said that wrong. Graham, this guy is really big time because Graham led to Graham staining, which we usually do in the lab when we're actually on campus. But we'll see if we can get some virtual Graham staining for you guys. Joseph Lister, you probably heard of Listerine. Came from him. Florence Nightingale, if you're going to be a nurse, you got to know Florence Nightingale. And then these guys, Jenner, von Behring, and Kitasato, I think I said that right. Ehrlich led to chemotherapy, so that's kind of big time. And then Fleming led to pharmaceutical microbiology. And these guys were all, like I said, in the golden age of microbiology between 1857 and 1907. So during those years, 4 important questions were being asked. Question 1, is spontaneous generation of microbial life possible? So they thought in the olden days, the before times, they thought that microbes could just spontaneously come about or generate out of nowhere. There was no seed or infection or anything. Microbes could just pop out of nowhere. And so they were asking, is that really possible? Well, we still drink a lot of wine, but winemakers, they were trying to ferment their grapes into wine. So they wanted to know what causes fermentation so that they could alleviate the bad fermentation processes and keep the good ones. Question 3, what causes disease? We're still looking into that. And the 4th question of the golden age of microbiology, how can we prevent infection and disease? 4 important questions. So we'll talk about those later on in the chapter. This is a diagram of the 3 major domains. So this, maybe you've seen these before, these, like, it's kind of like an evolutionary tree. So here's our primitive ancestor, evolution, 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 branching, evolution this way, evolution this way. And through millions of years of evolution, we came up with 3 domains. So bacteria, that's what we're mostly interested in this class. Archaea, they're microbes as well, but they're not really harmful to us, so we don't study them as much. And then eukarya, that's us. So we are eukaryotes. So Carolus Linnaeus, a Swedish botanist, so he's studying plants, he developed a classification system, and now we classify microbes into, like I just showed in the cartoon, bacteria, archaea, and eukarya. Bacteria are prokaryotes. That means they have no defined nucleus. Now this is tricky. They don't have a nucleus, and probably most of you know that in the nucleus is DNA. Well, bacteria do have DNA, but it's not in a defined nucleus with a phospholipid bilayer. So bacteria are prokaryotes. Archaea are prokaryotes as well. That means they have no defined nucleus. And then eukarya, that's eukaryotes, that's us. We have a defined nucleus where we keep our DNA. So eukarya includes not only us, but all the eukaryotic microbes. So fungi, for example, yeast and molds are fungi. Protozoa, so proto means pro and zoa means animals, so these are, a lot of them are single-celled animals. And then algae, they're eukaryotic. Eukaryotic disease causing small animals would be worms, eukaryotic worms or helminthes. And then viruses obviously are not small animals. They're unique. They have a unique classification. And we'll talk about viruses. The whole chapter has to do with viruses. So just don't be tricked by this. Prokaryotes are classified into the domains of archaea and bacteria only. Prokaryotes are not classified into the domain eukarya. So this is a stain, and you're looking at it on a microscope, put on a microscope slide of some cheek cells. So here's the nucleus of a cheek cell. Here's the nucleus of another cheek cell. Here's the kind of, I'll sort of outline this. Here's the plasma membrane of the one cheek cell. Oops, I don't work so good with this mouse. But here's another cheek cell. And then here's the bacteria, these little rod-shaped guys. They have infected the cheek cell. So let's kind of show you the difference in size in a eukaryotic cheek cell. So I think this is probably a human cheek cell. How big the human eukaryotic cell is and how small the bacterial prokaryotic cell is. Okay, so we already said this. Bacteria and archaea are unicellular prokaryotes. Well, maybe we didn't point out that bacteria and archaea are unicellular. We pointed out that they're prokaryotes, but we have to point out that they're unicellular as well. Anywhere there's moisture, you can find some species of bacteria. Bacteria have this cell wall. So our cells, human cells, eukaryotic cells, don't have cell walls. And plant cells have cell walls. Bacterial cells have cell walls, and there's this special molecule, peptidoglycan. So peptido, so that part of that molecule is a peptide or a protein, and glycan means sweet, so like glucose. So part of that molecule is a sugar. And this is really important. You guys should really buddy up to this next slide, because you'll see it, not only a direct question about the peptidoglycan in the cell walls, but also in the laboratory exercises, because it's that peptidoglycan in the cell wall that will pick up the stain when you're trying to stain your bacteria so you can see them. Archaea use other polymers. So polymer is just another name for this thing, peptidoglycan. A polymer means just many things stuck together, many proteins and sugars stuck together. So archaea use other polymers in their cell walls, not peptidoglycan. A eukaryotic plant uses cellulose in its cell wall. So you have peptidoglycan in the bacterial cell wall. You have cellulose in a plant cell wall. Cellulose is that stuff called roughage when you eat veggies and you have that roughage. It's good to eat. Or fiber. That's cellulose. And then archaea are often found in extreme environments like Yellowstone, which I've been to like 50 times because I grew up in southern Idaho. I love Yellowstone. I love looking at all the different colors that archaea produce in the hot springs. Most archaea don't cause disease. We don't study them as much. Archaea help degrade dead plants along with fungi, which all help to degrade dead things. So this is the cartoon I was talking about. You just make this your best friend. So this is let's start up here on the left side. This is a rod-shaped bacteria, and if you take this rectangular piece out of that bacteria and you enlarge it, you get this slice. And then what they do is they cut this little piece here out of the rectangle, and then they enlarge it over here so we can see what the bacterial cell wall is made of. Now, before I go on, let's differentiate. This is a gram-positive cell wall. There's gram-positive, so it'll stain positive. We'll talk more about gram staining later, but gram-positive, and then there's also gram-negative. So when you do your final lab, you'll have to, the first thing you'll do is you'll stain your bacteria because you don't know what it is. You'll stain it to see if it's gram-positive bacteria or gram-negative bacteria. So let's look at the cartoon of the cell wall. So this part here, this is all cell wall. It kind of looks like, you know, it kind of looks like a wall because these little things here, they look like bricks, don't they? So this is bacterial cell wall, and then deep to the bacterial cell wall is the phospholipid bilayer of the bacteria plasma membrane. So all these little blue circles here and the yellow tails that go with them, okay, and that's the phospholipid bilayer. And then there's these integral membrane proteins. So you have the bacteria with its plasma membrane, which is made of a phospholipid bilayer, and then on top of that you have the cell wall, and it's made of peptidoglycan with these acids, lipoteichoic and teichoic acid, stuck in the bacterial cell wall. And we'll go over this more in detail when we talk about the bacterial cell wall. Okay, so here's I'm not sure what kind of microscope they use to take this, but this is mold. It's where we get penicillin from. You can see there's spores, these little things here, and then these sort of, well, they're called hyphae. They're kind of like rods, so you go hyphae and then they're full of spores. That's a mold. Here's yeast. Probably everyone's heard of yeast. You use yeast to bake your bread, to make beer. This is a yeast cell, and then this little piece here is a bud. And this is Saccharomyces cerevisiae. So cerevisiae, we use that to make beer. So fungi include molds and yeast. They're not photosynthetic. That's important to remember. They are not plants. Molds and yeast are not plants. Fungi, in general, have all the regular eukaryotic organelles. So they have the nucleus, for example, the endoplasmic reticulum, the mitochondria, the Golgi apparatus, all those things you learned in previous classes. Along with some archaea, fungi do most degradation of dead plants and animals. Most degradation. Okay, and we talked about the molds and yeast. So we're still doing the microbial overview. Now we'll look at some eukaryotes. A eukaryote that is a unicellular microbe is called a protozoa. Remember I talked about this earlier? Proto means pre, zoa means animal. They're usually single-celled. So here's an amoeba, and it's a single cell. Here's the nucleus of that single cell. And then when it pushes its cytoplasm out into the periphery like this, those are called pseudopodia. Here's a Eukaryotes, a protozoa. It's got cilia, so cilia can move back and forth. We actually, our eukaryotic cells in our trachea have cilia as well. Here's a perineum. Parenema. And it's single-celled, so there's 3 parenema here. There's 3 of them, and they have flagella. So our humans and mammals, sperm cells, have a flagellum that allow them to move. So we said this protozoa have all the regular eukaryotic organelles. Most protozoa are motile. That means they can move. They have the pseudopodia, so we looked at that. We have the cilia. They call them hair-like structures. You only find cilia in eukaryotes, so don't be tricked on a test question. Do prokaryotes have cilia? No, they do not. Only eukaryotes have cilia. Flagella are longer than cilia, so you can kind of see that. So look, let's take a look. Here, if we measured, there's a cilia on Eukaryotes, here's another cilia, here's another cilia, and then we look at the flagellum on Parenema. Look how long that is. So flagella are longer than cilia. Flagella you can find in both prokaryotes and eukaryotes. So pros is just my shorthand for prokaryotes, and eus my shorthand for eukaryotes. So this is just a life cycle diagram of protozoa. They're unicellular microbes. So in the cyst stage of their life cycle, they're in a special structure, a cyst, and they can be passed from one individual to another in feces. They are resistant, so it's hard to destroy them, and they're infective. That means you can be infected by these unicellular protozoa when they are in their cyst stage. So then when they're in their cyst stage, to get to the next stage, they pass through this phase excistation. So they come out of their cyst and they form this thing called a trophozoite. This is usually when they're inside of you. When they're in this form, they can feed. They are motile. That means they can move, and they can replicate. So the feeding and growth portion of a protozoan's life cycle, so feeding and growth, so replication is growth, is known as the trophozoite stage. And then the cyst stage is the resistant stage. Probably everyone's heard of malaria. That's a protozoan, and it's called Plasmodium. Most protozoa are free living. It means you can find them in a lake. Some cause disease. Okay, now let's look at algae. So this is a algae called Spirogyra. I think there was a band in the 60s named Spirogyra, but I can't remember. Anyway, it's just a unicellular chain of cells. So, for example, here's one cell. And then, does anybody know what this thing is? What this is formula for? If you said, well, let me give you a clue. You take in CO2 and you make oxygen. If you said photosynthesis, you were right. So plants and algae take in CO2, combine with water, arrow goes this way, you make glucose, 6C6H12O6 plus oxygen, and then the plants give off oxygen. So algae, that's a eukaryotic cell, so it has all the eukaryotic organelles. So for example, mitochondria, Golgi, ER, etc., etc. Algae are photosynthetic. That's why I put this formula here, because they take in CO2 and they make oxygen. For us, they also make glucose for themselves. Algae provide most of the oxygen that we have on Earth. Unicellular algae, they're usually found in freshwater ponds. You can find them in lakes, oceans, and soil. And then seaweed like kelp are found. Obviously, if you've been to San Diego, you've seen that. Other microorganisms include a virus. So SEM, this means a scanning electron micrograph of a virus. We'll talk a whole bunch about viruses, but basically these things on the outside allow the virus to stick to your cells. Parasitic worms. This light micrograph, that's what LM stands for, light micrograph, of a parasitic worm found in a blood sample, probably a human blood sample. So here's the worm. It's the worm, and this is your red blood cell. Parasitic worms, they can be microscopic or they can be 23 feet long. Oh my Zeus, that's pretty long. Viruses, we'll talk a lot about viruses. Viruses are not cellular. They are obligatory parasites. That means in order for them to survive, they're obliged to be parasites in your cells. Viruses are composed of either DNA or RNA, and then they're surrounded by a protein coat. So this thing over here would be its protein coat. And coronavirus is an RNA virus.